and it's my immense pleasure to introduce Hugo Marchand from the National University of Mexico and the Institute of Neurobiology there, who will be talking about the neurophysiology of isochronic beat perception in primates. Hugo, the floor is yours.
Well, thank you. I'm really happy to see, good friends, including Annie and Sonja. And I'm delighted to be here and to talk a little bit about what we are doing now, at least some of what we are doing now. So I'm going to talk about brain dynamics in the primate audiomotor system during rhythmic timing. Well, what's going on? Here we go. So beat-based timing refers to the ability to detect the regular pulse or beat in music and to respond synchronously to the beat during dancing and musical ensemble playing. So as Annie was discussing in his talk, for a long time, beat perception and entrainment has been considered specific human traits. However, recent studies have shown that monkeys have the audiomotor machinery for regular pulse perception and entrainment. And here we have some papers related with this issue. In our lab, what we have done is to train monkeys in a synchronization task that started with a perception epoch, where animals place their free hand on a lever while brief stimuli were presented isochronically.
Sorry to bother you, Hugo, we're not seeing your screen, just in case you wanted to be sharing.
Oh, you are not seeing
no, I think you did not share, so maybe just quickly share your screen.
Oh, I'm sorry about that.
No, do not worry. I just wanted to not let you get too far in without letting you know.
Wait, where is this again? Share screen. I'm sorry. I thought it was sharing this. What about now? Can you see my screen?
Yes, thank you. And did you make sure you clicked on share sound when you shared screen, right?
Yes, yes.
Fantastic. Okay, you're all ready to go. Sorry about that.
Okay, sorry. So let me just go again. This is a scheme of our synchronization task. So the monkey perceives, isochronic stimuli while not moving, and then after 3 intervals, starts to synchronously push a button with the metronome. And if the time relation between the stimuli and the tap is below an error threshold, the monkey receives a reward in form of some drops of juice. We use target intervals that go from 450 all the way to 950 milliseconds that change from trial to trial. It's a very dynamic task. And we use auditory or visual metronomes. We also have a modification in this task in which after synchronization, we have a continuation epoch in which the monkey taps, isochronically, but using only some sort of internal timer because there is no stimuli. Okay, so how we quantify the behavior of the monkeys? What we do initially is to measure the time relationship between the stimuli and the taps, and those are called asynchronous, and we can plot them in a polar plot where 0 means a perfect synchronization between the stimulus and the tap. And what you can see is the distribution of the asynchronous of the monkeys are around 0, with some of them in negative values, which is similar to what you see in humans. So we can say that monkeys are capable of temporal anticipation. They can extract prospective information from regularly stimuli in order to plan and coordinate anticipatory movements. So here is a video of a monkey doing the task. So presses the lever and then synchronizes. You can see that there is a very nice timing between the monkey pressing the device and the auditory pulse. So in addition of the asynchronous that allows you to measure the predictive behavior of the monkeys, we also have trial-by-trial information about the temporal variability that shows, an increase as a function of time, which is the scalar property of timing. And we also have the constant error as a measure of timing accuracy. So what we have been doing in the lab is to record from what we call the main core timing network that includes the supplementary motor cortex, the basal ganglia, and the motor thalamus, that we hypothesize is a main core timing network for a series of different timing tasks, including beat perception and entrainment, and that these structures are interacting with context-specific structures to deliver a particular timing signature in behavior. And one of the things that we have done then is to record from supplementary motor cortex. My talk is going to be mainly about this area. And we have recorded this across different tapping tasks. So initially what we found is that neurons in this area are tuned to the tempo of the metronome. So they have a preferred interval where they have a larger discharge rate for a particular duration. And what we have found is that across population, all the tested durations are represented. In addition, we also found that neurons are tuned to the serial order of our sequential task. So this is the synchronization continuation task, where we have 3 synchronization intervals and then 3 continuation intervals. And you can see that there are neurons that prefer different serial order elements, like the neuron in B shown here, that responds for the 5th element of the serial order, but also is tuned for longer durations. So it means that the supplementary motor cortex has a representation of the temporal and serial order elements of our task, and that this is dynamically represented. In fact, when we plotted neurons that were tuned to serial order or to duration, and we order them based on their peak of activity, what you can see is a neural sequence, some sort of dynamic process where the neurons are evolving and passing information across the task. And so to try to characterize these properly, you need to use dimensional reduction techniques that allows you to measure how these populations of neurons are encoding in their population information. So, you know, now what the people are using is PCA techniques to project time-varying population activity into a low-dimensional state space that allows you then to study the neural responses as a dynamical system. And in fact, what we found when we used these analytical techniques was that in SMA and pre-SMA, we have neural trajectories in state space that are cyclical, where each produced interval in the rhythmic sequence is represented as one circular loop. Crucially, longer produced intervals produce larger amplitude. So in this case, the red are trajectories for our longer tempo, the slower tempo, and the green trajectories are for the fastest tempo. So, and this happens for both the synchronization and the continuation epoch of our task. So in fact, when we measure the radius of these trajectories, you can see a monotonic increase in the radius as a function of target interval, while the linear speed of these trajectories is pretty much constant. So an important part of these neural trajectories is that they behave as what we call periodic tangent circles with different diameters. So this is short durations, this is long durations, but they are converging in an attractor that corresponds to the moment in which the monkey is tapping. So to try to characterize this quantitatively, what we did is to measure the distance between a constant point and the point in the position of the trajectory where the taps occurred, here in green. And you can see that this distance is constant across durations. In contrast, when we measure this distance but for the halfway of the trajectories, you can see a monotonic increase. So therefore, what we are suggesting is that these results suppose the idea that neural trajectories behave as regenerating tangent circles that encode the duo between taps in the amplitude of the trajectories. And at the same time, they trigger the internal beat signal once the neural dynamics reach this attractor zone. So, you know, some time ago with my friend Heng-Jan Honing, we suggested the gradual audiomotor evolutionary hypothesis that basically suggests that beat perception and entrainment has emerged gradually in the primate order. So the incredible human flexibility to entrain to complex beats depends on the close interaction between the auditory and the premotor areas. And what we are suggesting is that the monkey has all the brain machinery in this audiomotor circuit to perceive and entrain the simplest beat, a regular metronome. So to test this, what we did is to record then in pre-SMA and SMA and compared auditory versus visual metronomes. So do we have a model timer, or there are some characteristics between the auditory and the visual metronomes that shape the neural trajectories? So, and then here I'm showing, you know, these neural trajectories for 2 intervals in an auditory metronome and 2 intervals in the visual metronome. And you can see that these trajectories are again circular. Each loop represents 1 produced interval. And here in green, we have the tapping times. So what you can see is that when we compute the subspaces for duration, for tapping, and for modality in these multidimensional neural trajectories, we found specific traits that are important. The duration manifold, you know, has a clear increase in a diameter for longer durations with respect to shorter durations in both modalities. So this is light blue for short, this is dark blue for long in the auditory modality. So if you look for the subspace where the tapping is occurring, you found then the attractor for the tapping. And this occurs for every tap in any modality and in any duration. So it means that probably, you know, the fact that these neural trajectories are reaching this attractor, that moment means this is the moment in which we have an internal pulse representation. It doesn't matter the tempo, and it doesn't matter the modality. However, we also found in these neural trajectories that there is some covariation between modalities. However, there is also some differences. So if you compute the difference in the angles between these manifolds, the auditory and the visual, you can see that there is some degrees of differences suggesting that there is a partial overlapping representation of time between auditory and visual in the supplementary motor cortex. So now we are developing an experiment where we record simultaneously in different key brain areas during beat perception and entrainment in order to determine key bottom-up and top-down processing associated with these cognitive abilities. And specifically, we are, you know, interested in understanding the pure auditory processing. What about the attention elements? What about the context-dependent auditory processing during beat perception and entrainment, the error prediction signals, and of course, the predictive beat signal interacting across the circuit. So for that, what we have done is to record in multiple areas, including the dorsal auditory areas, including the core auditory and the belt auditory areas of this dorsal stream that Annie Patel already mentioned. Of course, we recorded in PSMA, which we suggest that is the internal timing beat structure, but also the output in terms of the primary motor cortex and the putamen, and an intermediate area that is an association area, it's area 7A, that receive projections in the monkey from the belt, the dorsal belt area. And then with this analysis, then we're going to be able to determine the neurophysiological mechanism behind the passive auditory sensation, the tapping movement encoders, the active sensory encoders, the internal beat cells, and the error prediction signals. And so what I'm going to show now is a preliminary analysis on these large databases for one monkey. And what we have done is an initial analytical approach in which we use a time warping analysis that allows you first to determine whether the activity of cells was better aligned to the sensory, in this case, the metronome, or the motom, the tapping events. And this is based on the statistics of the responses of cells across trials aligned to both events. So in addition, we characterize the profile of activation of neurons using what is called the Poisson train analysis, that basically what it does is identifies the response onset of cells and the duration of the response using the interspike interval histogram. So here I'm showing the activation profile of one single neuron in the core A1 area during the perception epoch that where the monkey is not moving, is just perceiving the metronome. And then during the synchronization epoch, where it's synchronizing to these isochronous poles. You can see that these neurons have a very strong response just a few milliseconds after each tone, and that they don't have any, they don't distinguish between perception and synchronization. And in fact, when we recorded this neuron in a passive situation where the monkey is not allowed to move and doesn't receive a reward, the responses are very similar, suggesting that these neurons are related with sensation, just with the input. So when we analyze this at the population level, so here we have cells, and each line is one single cell. Each circle represents the peak activity for each neuron. You can see a large population of neurons here in green that responding very stereotypically across all the stimuli with short latencies. And this is really clear when we plot the peak activity in a polar response where 0 is the stimulus. You can see that there is a large population of neurons that are responding just after the stimulus, and this response is very similar in the passive situation. However, when we recorded in the belt area, we found different types of responses. First, this is another neuron that responds differentially between the perception epoch versus the synchronization epoch, with a larger discharge rate in the perception and a modulation in the passive situation. So when we analyze these at the population level, you can see that the latency of response in the belt area is larger than in the core. And you can see also population of neurons that are responding at intermediate durations, including some that are responding before the next stimulus. And this is quite clear when we compute, we project the peak responses in the polar plot, where you can see a complex response across the whole interval and modulations between the synchronization and the passive situation. So however, you also find something that I found really interesting. These are cells that are classified based on our warping analysis as motor cells. These are much better aligned responses to the tap rather than the stimulus, and they occur in the belt area pretty close to the neurons that are responding to the stimuli. So here is an example of a raster of this neuron. This neuron responds during synchronization, not during perception. They don't respond in the passive, and they are pretty well aligned to the tapping events instead to the stimulus events. And when we analyze this at the level of the population, you can see large numbers of cells responding after each tap and large number of cells responding before, like predicting the next tap. And where we interpreting these responses as some sort of top-down pulse signal, probably coming from motor areas. So you can see that these neurons have a very nice alignment to the tap, and some of them are predicting the next tap. And there is few responsive in the passive situation. Finally, we found, similar responses of what we already described in pre-SMA, which means neurons that are attuned to durations, to serial order, and at the population level, we see these moving bumps where the neurons are representing information across all the duration. In fact, when we do the polar plots on these neural sequences, what you can see is a structural response across the whole interval, and then a large groups of cells responding just before the tap. And the hypothesis that we have is that this group of cells are the ones that are related with the attractor of the pulse representation that I showed for the neural trajectories. And of course, when we test the passive situation in these neurons, there is nothing. So I would like to finish saying thank you, especially to Yaneri Ayala that is here, Germán Mendoza, Juan Pablo Mendez, and Luis Prado that have been really involved in these new experiments that are really challenging technically and analytically. And also, I would like to thank all the funding. And I would like to say gracias por su atención.
Thanks so much, Hugo, and much appreciated that you kept time so well. Lovely, lovely results. I like the sophistication of this network approach. So this is really quite intriguing. And I want to make sure I leave the floor open to some questions. Let me have a look. They will be coming in. There's always a bit of a delay.
Yes, of course, I would like to say that this is preliminary, and these are results from one monkey. And of course, this is just the beginning of a humongous enterprise to try to understand how the dynamics between areas is happening and how the flux of information, of the auditory side and the flux of information of the internal beat representation are, interacting. But I think we are in good shape to try to answer some of these questions.
All right, and here they come, if I may. Peter Keller asks, is there anything interesting happening in the interval after the final tap, like evidence for self-sustained activity in the case of the top-down component?
So, but you mean after the tap or after the tap?
It's after the final tap.
Okay, well, the first thing that we find and this is really something interesting across all the areas that we have recorded, including the auditory areas, is a strong signal related with word. Okay, so correct trials, there is a super strong signal every time that the monkey receives a reward. And when there is an incorrect trial, there is nothing. Okay, and then what we are testing now is whether the magnitude of reward is modulating the responses. It occurs in the core area. It's a very strong signal. Now, in the intertrial epoch, we have not looked at whether there's modulations on activity based on the prior trial, on the prior duration, but we will look. So I don't have an answer yet to that.
Okay, and I see a lot of clapping. I see Henk-Jan's comment, great Hugo, preliminary results, but very nice. This is all very promising. I wondered, in light of the prior talk by Annie, you very distinctly talk about the pulse. And of course, we can potentially ask the question, could we think about some form of a continuum hypothesis here as well? That with more increased capacity to motorically learn and synchronize, we move from the pulse to the beat. I'm just trying to think whether we're still missing out on some connection here.
Well, I think this is a primary fingerprint of an internal pulse representation, in the medial premotor areas. I think probably shared between species, that can follow at least an isochronous beat. What's going to happen when you have more complex stimuli with a metric structure? I'm not sure, and I'm not sure whether this is going to hold for all of them. However, well, this is a testing hypothesis, right? And now that we have the ability to record in these areas in humans, we're going to be able to test these hypotheses. And I think we have a very clear result in the monkey that can be tested.
There is one more question, Sonia. I don't know if you see.
Trying to roll. It looks more like statements, but maybe I'm overlooking something.
Henk-Jan says he wants to know whether...
sorry, Henk-Jan. Go ahead, Merle, you can raise it.
He just says Annie's and our own JE hypothesis suggests the parietal areas are to be involved as well. Do you have or make a plan to take measurements there as well?
We did.
Yeah.
I think I mentioned that. Area 7A is an area in the posterior parietal cortex of monkeys that has a very interesting anatomical properties, because it's a visual area, but it also receives, in where we recorded, projections from the posterior belt area, and also projects to the premotor system in the frontal lobe. And we have recorded there, and we found, I didn't show those results, but we found neurons that are behaving similarly to what we found in pre-SMA. At least that's a preliminary observation. And of course, we will look at different properties there. So of course, we are very interested in this area.